Figuring out how certain genes are involved in cancer is an important part of understanding and controlling these diseases. Some genes are expressed in embryos and tumors, but not in normal adult tissue. These oncofetal genes can be used as biomarkers and might be good targets for cancer therapies. Saja Fakhr AlDeen began studying an oncogene linked with breast cancer when she was a graduate student supported by the Kuwait Foundation for the Advancement of Sciences. The gene, called CRDBP, was known to regulate the activity of other cancer-related genes. Fakhr AlDeen discovered that CRDBP isn't really an oncofetal gene. Her team detected CRDBP expression in normal adult breast tissue. But in these tissues, CRDBP produces a shorter form of its protein with one end missing. Antibodies used in earlier studies could only bind to the full-length protein, so the shorter form went undetected. Even though it isn't an oncofetal gene, CRDBP is still important in breast cancer. Fakhr AlDeen showed that breast cancer cells lacking CRDBP weren't as good at surviving and proliferating as normal cancer cells. She and her biomedical engineering colleagues also worked on refining a marketable system to extract molecular complexes of RNA and proteins. Using that tool, they identified RNAs that CRDBP interacts with, giving them a list of genes affected by CRDBP. Next, she plans to investigate the genes on that list in order to understand how CRDBP is involved in breast cancer. In the long run, Fakhr AlDeen hopes that CRDBP or one of its downstream targets will turn out to be an effective target for breast cancer therapy.